So, hi, hello everyone, welcome to Raaonline. This is Dr. Srivatsan, your faculty for biochemistry, and in today's session we will be seeing about eicosanoids. Okay, so eicosanoids, what are they and how they are different from hormones? So, these actually acts as a local regulator. In some books they refer it to as local hormones. But this should not be considered as an hormone. Why? Because hormones are the ones which is being synthesized in the specific endocrine glands, and this will get stored in those glands itself, and whenever there is a requirement, this gland will secrete this hormone in the blood, and this circulates in the entire body. Okay, and it goes to various kinds of tissues which are very farther away or very too farther away from the presence of the main gland. Okay, but in case of eicosanoids, these are nothing but local regulators. So what it does, it regulates the thing. Okay, they get synthesized at a specific point, at a specific cell, and they help as a mediator or they help as a regulator in that same, near the same cell itself. It won't go to any farther away place, unlike hormones. Hence these are more rightly called as regulators, local regulators. Now these icosa, what do you mean by icosa? Icosa are nothing but 20 carbon. Okay, icosa means 20. So it means these are derivatives of 20 carbon containing what acid? Arachidonic acid. So these are nothing but a derivative of 20 carbon containing arachidonic acid. So how do you get arachidonic acid in your body? Arachidonic acid is a semi-essential acid, means it can be consumed through food. At the same time, this can be synthesized partly by your body itself from linoleic acid. So from linoleic acid, you get to have arachidonic acid. But in case of prostaglandin synthesis or in case of various kinds of eicosanoid synthesis, what happens? The main source of your arachidonic acid will be your phospholipid bilayer or phospholipid cell membrane. Okay, so consider this as a leukocytic cell. So consider this as a leukocytic cell. So this, if you enlarge this part, so it will be having what? It will be having various kinds of phospholipid bilayer. So this is the phospholipid bilayer. Now, if this phospholipid bilayer gets acted upon by phospholipase A2, A here stands for arachidonic acid. So if this gets acted upon by phospholipase A2, they form arachidonic acid. So what is the shape of this arachidonic acid? It's a very interesting shape. Here we have got the carboxyl group, COOH group, and we have got 4 double bonds in arachidonic acid. So this is the structure of arachidonic acid, guys. Now this arachidonic acid, they can combine, they have got various different kinds of fates. Okay, they can enter into various kinds of pathways, and this pathway will be determined by the amount of, the cell where this arachidonic acid synthesis, the cell where this eicosanoid synthesis is being taken place. So, it depends upon the tissue, or it depends upon the organ, or it depends upon the cell where this kind of reaction takes place. So, accordingly the pathways differ. So, I am going to present here all the pathways together so that you will be having an overall picture. Now, the first one, okay, this can enter into COX pathway, which is nothing but cyclooxygenase pathway, or this can enter lipooxygenase pathway, or this arachidonic acid can combine with lyso PAF. So, what is this PAF? It is nothing but platelet activating factor. So, this can get combined with lysoplatelet activating factor to form what? Platelet activating factor. So, this usually occur where? In platelets, in monocytes, in macrophages, in endothelial cells,